Okay, so good morning and welcome to this innovation workshop on the day 2 of Genesis 2021 conference. My name is Alicia Gallier. I am Business Development Manager at One Nucleus, and I'm delighted to welcome you to this innovation workshop this morning. So this session is in collaboration with Collaborative Drug Discovery, and I'm delighted to have Susana Tomasio, who's going to be our speaker and host for the session. So Susana is going to kick off with a short introduction via some slides. And then very exciting, we have a live demo on their Drug Discovery Vault platform. So I'm really looking forward to seeing and hearing more about this software. Just a few logistical points. So the chat function is enabled. Just catching up with Susana before the session. I'm very happy to make this an interactive session, so please don't be shy and please drop your comments and questions into the chat and we can pick those up along the way. Please remain muted, much appreciated. And as you will have heard, we are recording the session, so it will be featured as part of the conference on demand content, so you can always catch up on it afterwards. So not much more from me. So Susana, I'm going to hand the floor to you. Please feel free to share your screen and away you go. Thank you.
Perfect. Thank you, Alicia. So hi, everyone. Just starting to share my screen here. Can everyone see my screen?
Looks good.
Yeah. Okay, perfect. So yeah, thank you. And it's a pleasure to be here today. My name is Susana Tomasio. I'm a senior application scientist at Collaborative Drug Discovery, and I provide technical support for CDD customers all over the world. My background is in computational chemistry, and I've been with CDD for over 3 years now. And today I will be giving a live demo of the CDD Vault platform, and I will show you how this modern application can be used to register molecules, biological entities, and all the assay data associated with your experiments, allowing you to organize your data in a simple to use and secure system. So just before jumping into the live demo of CDD Vault, I would like to mention that CDD Vault has 17 years of experience in storing, managing, and organizing chemical and biological data in a collaborative environment. It is a flexible web platform which you can use to store your small molecules, your antibodies, your proteins, all your biological entities, along with the assay data. It can be seen as your own single source of truth, where all your internal and external collaborators can access through an easy-to-use web interface. So this slide shows how CDD Vault can be used to create and configure seamless registration of entities by setting up different vaults. Basically, depending on the types of entities that you currently use in your own research, you can have different vaults to store these types of entities. And this is what will be the focus of the workshop today. Just before we go there, I just would like to show you that these are some of the examples of the customers that use CDD Vault. Okay, without going further, I think I'll just have to stop sharing one more time, and then I'm going to share my screen here again. And perfect. So I guess everyone can see my screen.
That looks good. Yep.
Perfect. Thank you for confirming. And so this is our website, collaborativedrug.com. And as mentioned earlier, CDD Vault is a web platform. There is no need to install any software in your computer. All you need is modern browser and internet connection. CDD Vault comprises 4 modules: an electronic lab notebook, which allows scientists to register, to write in a flexible way all the results of their experiments. What I really like about this is that it's fully integrated with the activity and registration module, which means that you can be working in your electronic lab notebook and you can register all your entities and assay data directly from the ELN. We also have an inventory module, which allows users to keep track of the quantity of their batch compounds or entities. And we have a visualization tool, which allows to to plot, filter, and mine the data. So basically, when we log in using the email address and password, so we reach this page where we just type in our email address and password, and then we reach this landing page where we can see the name of the vault. And as mentioned in my slide, you can use CDD Vault to register different types of entities. In this particular vault, I'm using it to store ADCs, so antibody drug conjugates. But if you see here on this drop down, I am a member of all these other vaults. So I also have a vault to register small molecules, oligos, reagents, payloads, and so on. So depending on the types of entities that you are using in your research, you can have different vaults to store all of those entities. The advantage of all this is that they are all connected, as we will see in a moment. So when we log in, we see the name of the vault, we see the name of the username along with the user role. I am the vault administrator of this platform, and therefore I can add other members into this database. When I add a member into the database, I can choose which user role I want to assign to that new member. So really depending on what I want to allow them to do, I can decide which user role I want to assign to this new member. Also, the key concept in CDD Vault is the concept of a project or projects. Basically, all data and all entities registered in the vault must be associated with the project. And then each project can have different members, and the members will only be able to view the data of the project that they are a part of. So this allows users or scientists to have their own internal and external collaborators within the same database. Okay, so let's go here all the way down into a search results table where I am showing an example of the types of data that you can store in your private secure vaults. As mentioned before, this vault that I'm showing at the moment is a vault to store ADCs, antibody drug conjugates, which we can see here on the left-hand side in this column. Whenever we register either the sequence or the synonym or a unique identifier, CDD will automatically generate a unique ID, which comprises a prefix, which the vault administrator chooses, and a sequential number. Then we have a few sections or a few columns in here. We have the entity fields and the batch fields. These fields are created and customized by the vault administrator. So depending on your own business rules, depending on the types of information that you need to store or that you need, depending on the types of information that you need to associate to your entity, the vault administrator can create these fields. So the entity fields will have all the information which is common to the batches of the same entity, while the batch fields will have all the information which is specific for each batch. And here, these fields, they can be text, they can be an image, they can be number, they can be an URL, as we can see in here, but very importantly, they can also be a link, right, to another entity registered on a separate vault. So as we know, an antibody drug conjugate comprises an antibody, a linker, maybe one or two linkers, and a payload. And what we have in here is the links to those individual entities. So I can simply follow this link. For example, if I want to take a look at the antibody information, I can simply open in a new tab. And I am now in my antibody vault, and I can see all the information for this particular antibody. I can see the sequence and all the information for this particular antibody. So going back into the search results table, we also have the links into the oligos or to the linkers and the payloads. So they are all in separate vaults. because they are different entities. Also, whenever we reference a particular entity in the electronic lab notebook, CDD will automatically generate these links to the particular experiment. And this is why I really like this integration of the registration in CDD Vault and the electronic lab notebook, because in this case, as you can see in here, it is telling us that this batch of this ADC is linked in this ELN entry. I can also open in a new tab, and then I can view the content of this electronic lab notebook experiment. So I can see the title, and then I can see all the information. And this is where I registered my ADC, and that's why it's linked in the search results table. Again, I can also view the information of this particular ADC. And this is what we call the entity page, where we can see the entity ID or entity name, which is automatically generated when a new structure or sequence or synonym is registered in the database. In the overview page, I can see all the fields generated by the Vault administrator. And then in the batch fields, I can view, I can see all the information, again, that is specific for this particular batch. We can see the link into the ELN experiment because this particular batch was registered from the ELN experiment. And that's why we have this link. So that's the advantage of registering data and entities from the electronic lab notebook. At this level, we can also view the inventory. Basically, we can register an initial amount for your entity. You can have a field for the units. And then each time a debit is performed, CDD will automatically calculate the current amount and update the location. All these other tabs have information for this particular entity. So the links will show all the ELN experiments or places where the entity was associated or linked. In this example, as we saw earlier, this particular entity was referenced from this particular ELN experiment. If your entity was tested in any plate or plates, we will show this information in here. And then in the assays subtab, you can see all the assay data that was performed on that particular entity. In this case, this ADC was tested in this cytotoxicity assay, and I can click on this link and I can view which batch was tested. I can view the run date, and then I can view all the readouts for this particular assay. Also, I'd like to mention projects. So as we saw, as I mentioned earlier, the key concept in CDD is the concept of a project, and you can actually share the access between projects. So for example, batch number 1 of this ADC is shared in these 2 projects. However, I have a 3rd project for my CRO, which is not in here, which means that whenever a member from my CRO project logs into CDD Vault, they will not be able to view this particular ADC or assay data. Okay, so I'm now just, we are going back into our search results table because we've done here a small detour. So this is the search results table that I was showing you, where we can see all the ADCs on the left side and the entity fields and batch fields, as we saw in more detail in the entity page. The next concept that I would like to talk about is the concept of a protocol or assay. So in here, in this search results table, we can see that all these ADCs were tested in this cytotoxicity assay, and the concept of assay or protocol in CDD Vault is used to store the data that you get from your experiments. So this can be experimental data, or even in silico data. So we pre-create the containers to store all the assay data that we get for particular entities, and then we import the data associated to those tested entities. So as you can see in here, this is a dose response protocol or assay where the data was imported in a raw state, and then CDD Vault performed the normalization and then calculated an EC50 and performed this dose response curve. These dose response curves, they are live in the sense that we can click in flag outliers and override, and then a new window pops up. And then in here, I can see on the left-hand side the legend. So basically, I am plotting 2 runs of the same assay. And basically in here, I can flag points that I don't want to include in the curve fitting. So for example, if I don't want to include these 2 points, I can simply click on those points. And then as I click Save the changes, the curve is being regenerated without those 2 points. The plot is also updated in the search results table, and then the IC50 and EC50 is also recalculated. As we can see in here, when I hover over the number, we can see that the value was calculated excluding the outliers. Of course, that I can always come back and re-add those points. We are not deleting any data. We are just flagging those points. So similarly, because this run was imported from an ELN entry, CDD will automatically give you the ELN experiment link. Here I can open this in a new tab, and again I can view my ELN experiment and all the details. We have here the assay annotation in this ELN experiment, which is this pencil shape icon in here, which basically keeps a summary of what was done in the assay. But basically, because the data was imported from the ELN, that's why we got that particular link in the search results table. Okay, so whenever we have a search results table like this, we can always click in Customize your report. And then here we can see all the fields that exist in CDD Vault. And then you can select which fields you want to see or not in that particular table. Basically, you can cherry pick which fields you want to see. Okay, so maybe just adding here the raw data as well. And then as we click Update the report, the extra fields are being re-added into the search results table. Okay, so moving on. So this was kind of an example of the types of data that we can store in your private secure vaults. All the entities will be shown in this tab in here. And then the next concept that I would like to talk about is the concept of protocols or assays. As mentioned before, the assays are used to store all the data from your experiments. So going into the assays, here we can see the assays that I have in this vault for the projects that I have selected. Just clicking, for example, on this first assay, we can see that all the data is organized by a run date. So each project will have different run dates. And then in the assay details, this is where we can, where we will give a name of the protocol. We can have a small set of categories. So we'll have a category, and then we can search for all the assays of a given category. And then the description, this is optional. You can, some users like to add the description of all the assays. Some users just like to give a small summary, but in essence, these fields here, they are called the metadata fields, and they can be customized. If there are additional fields that you want to capture for all your assays, they can be added as well, and the Vault administrator can customize those fields that appear in here. But the most important part of the assay or protocol is this section, the readout definitions. Let's go into edit mode, and this is where we can pre-create a series of readouts that match the data that you want to store. Plus, CDD Vault gives you the ability to perform custom calculations and perform EC50, IC50, or other type of intersection. So the first readout is the simplest one, is just readout definition. Basically, if you need to store a number, you choose the data type number. If it's text, you choose text. So you have here all the different data types depending on what you have in your Excel spreadsheet. So you'll just give it a name and then you choose the data type. The second type of readout definition is a calculated readout definition. And here the customers or scientists have the ability to perform a custom calculation. So clicking in the calculated readout definition, again, you need to add a name, the units, and then we have here this question mark, which is quite useful because it shows some examples of the types of formulas that you can perform in CDD Vault. Plus, it gives you the common functions that are available and how to reference the readout definitions, chemical properties, or assay conditions. So for example, going here into the formula box, if I want to perform an average, I just need to type A and we get a drop down with all the functions that have the letter A. So I just choose the average. And now to reference the readout definitions, it says here just to open a square bracket. So when I open the square bracket, we get a drop down of all the readouts that not only exist in this current protocol, but also in the other protocols of the Vault. If I want an average of my EC50, I simply choose the EC50 readout and then the formula is ready. Whenever we perform an average or a geomean or a median, you'll get this option here to choose the aggregation scope. And this is quite useful because, for example, the aggregation by batch and run, it's useful when in the same run you have replicates. The aggregation by batch and assay will basically average all the runs of your assay. And this is quite useful as well, because whenever we import a new run into CDD Vault, CDD will automatically update the average because you have the aggregation by batch and assay. The third type of aggregation is the widest one or the less specific, because basically this will also aggregate all the batches of your entity. In this case, it's the ADC, so it would aggregate, if there are more than one batch, it would aggregate those as well. So this is for the custom calculations. And then finally, we have the ability to perform dose response calculations and plots. CDD Vault uses the Levenberg-Marquardt algorithm, which is a 4-parameter logistic curve. And then here you can simply add the concentration, what you are measuring. Is this % of inhibition, or in this case, it's the cell viability. Whether your data is already normalized or not, CDD Vault can also normalize your data if you have the controls in the same plate or in a separate plate, or it may be that you don't have any controls, but then you can choose here the data calculation. You can choose the type of intersection. You can perform a custom intersection, let's say an EC60, for example, and you can have as many data calculations as you wish. And this is for the dose response calculation. Of course, that if your data comes in a plate and you have controls in the plate, we need to come into the control layouts. And then here we just choose the type of plate. We click in edit this layout. And then here you can add where the positive, negative, and reference molecules are in the plate. So let's say that I have a positive control on column 1. I just do one click, and now column 1 is marked as my positive control. Let's say that on column 4, I have a negative control. So I just click 2. times. And then for the reference or benchmark molecules, we just click 3 times. So 1, 2, 3. Once we save the changes, CDD Vault will know that in these wells, there are the controls and reference molecules, and the normalization will be based on this layout. Of course, that this also works in individual cells or wells and across the rows. Okay, so this is basically how we set up a protocol. So coming into the Explore Data tab, basically, once we have our data and entities registered, and bear in mind that we can register one entity manually, or we can simply use the Import Data tab where we can register multiple ADCs in bulk or other types of entities, depending on what you are using your Vault for. But really the advantage of having the data, the entities and assay data registered in a structured way is for searchability. So once I register my ADCs, once I register my assays, I can now perform a query search. I can decide which projects I want to search in. So maybe I don't want to search in my CRO project. And then I can search across the assays. I can search in any run or a specific run or runs in the past 7 days or 30 days. If the assay was performed in different cells, we can also choose if I just want to view the data for a particular cell and even a particular readout definition. So maybe I just want to view the ADCs that had a particular EC50. So this is all customizable. Of course, that if we are using a Vault to store small molecules, we can also perform a substructure search. And we can also select some of the properties, and we can kind of add some restrictions here for the type of data that we want to see. So depending on the fields, you can just say, oh, I want to view the data for a particular antibody or for a particular linker. So let's click search. So I'm just searching for the data in this assay. And here we just get a search results table similar to the one that I showed you earlier. And as we saw earlier, we can click in customize the report and just cherry pick the fields that we want to see. When we have a search results table like this, we can save the search. And the advantage of saving a search is, of course, if you have a complex query, you can just create your saved search, and then each week or each month, you can just rerun a saved search and check if there is more data in the Vault. A saved search is dynamic in the sense that if more data is added into the Vault that satisfy the query, then when I rerun a saved search, it will retrieve more data. And the saved search has its own home or its own tab in here. And similarly, we can also add into a collection. A collection is fixed, is a subset of entities or compounds that we can use to share with the members of our project. And the collection is fixed unless we update an existing collection. So if in your daily research or regular, you need to share data or share entities or small molecules with your colleagues, instead of sending an Excel spreadsheet with those, you can simply share a collection. Again, it's a secure way of sharing data. Collections also have its own home in here, and we can simply just share the collection. Of course, we can always export our data into Excel, CSV, or SDF file. Okay, also still on the topic of searching, let me just go here into this. So here I'm on my antibody vault, which I use to search, to store my antibodies. And you can also, of course, search for a particular sequence. So I have copied the sequence earlier on. And here, if I just search for a particular sequence of an antibody, CDD can also easily find that sequence. And then you can click on the antibody entity, and again, you can view the information for that particular antibody. So in this case, I'm on my antibody vault, as you can see here, that I can switch between different vaults. And as we saw earlier on the ADC vault, we were linking the ADC with the different parts of the ADC, and one of them is linked to the antibody entities. Okay, so we've been through the structure side of the vault, where we can register any type, any entity type. Maybe I haven't talked too much about small molecules today, but of course that CDD Vault can also be used to store small molecules. And we've seen that we can store either small molecules or any type of entity. We can store all the assay data in the structure side of the vault. And what I would like to talk about now is about the ELN. So the electronic lab notebook is fully integrated with the activity and registration system, but that's the place where we can write in a flexible way. Just imagine your paper notebook when you open a new page and you have a blank page, and then you can write in a flexible way. That's exactly what we can do in the electronic lab notebook. So here, if I just create a new ELN entry, right? So I'm just going to select a project where I want to add my ELN experiment, and I'm going to begin editing. And then what we get is this, a blank page. So I can add my title in here for my experiment. These fields here that you can see, they are created also by the Vault administrator. So again, the Vault is flexible enough in the way that the Vault administrator can decide which fields you need to have at the top of the experiment. And then we have this blank page where we can write in a flexible way. We have text, different colors, you know, what you would expect from a text editor. I mean, this is fine, but usually scientists prefer to have a place to start from, so a starting point. So the workflow that we usually recommend, I'm going to describe it now, is you can have a project for the ELN templates. And in this project, we have templates for the types of experiments that you currently run. And then in each of these templates, we have the sections that we want to make sure that everyone fills in whenever we perform that experiment. So let's say that today I go into the lab, I'm performing a screening. My workflow can be, I log into my CDD Vault, I come into this ELN tab, I select the ELN templates, and then from this list, I choose the appropriate template. So I'm performing a screening, I'm opening this template. In this template, I have all the instructions of what I need to do, along with a template for the Excel spreadsheet. So what I need to do now is just duplicate this template, and it's performing a copy of the ELN experiment. Now I can simply change the title, of course. So I'm screening, I don't know, XYZ, and then I can move this into the appropriate project. Now I can follow, so now it's in the appropriate project where I should be working on, and now I can simply follow the instructions. I can delete the sections that I don't need, and then I can simply follow the instructions in here. I can add a timestamp as requested. This timestamp will capture the local time, the date and the local time as defined on my user settings. And then once finished, I can simply finalize it, assign a witness, and then the witness will receive an email to check with a link to check my experiment. Once this is accepted, then the experiment will be finalized. If after finalizing the experiment, if there are any errors, so let's say that I made a mistake on my experiment and this needs to be corrected, the Vault administrator can always reopen a finalized experiment. However, of course, this will all be tracked in the audit trail. The audit trail, as you can see in here, keeps track of when the experiment was created, when it was modified, when it was assigned for witnessing, and if it was reopened after being finalized, it will also appear in here. So this is the workflow that we usually recommend to users, is to have different templates for their experiments, and then that way you know that everyone will be following that template and adding all the sections that are required. So just in terms of functionality, I'm just going to show you here an example of an experiment which pretty much shows all the functionality in the ELN. So it's not a real experiment, but it's really just to show, all the types of text that you can have, these checklists, the timestamp, images. You can simply drag and drop the images, or you can use the insert file attachment. Tables, of course, they are captured. PDB structures or crystal structures or Mol2 files, if you are working with those, you can simply add them, and then you have this really nice visualizer. I particularly like this because we can view the 3D model of the protein and the ligand at the binding site, and that works with Mol2 files as well. Of course, if you're working with small molecules, you can use this insert structure or reaction. You can draw the molecules, you can register them straight into the Vault. You see here that I added this molecule. It's not on my Vault because I don't see the ID yet. And therefore I can click register and register that straight into the structure side of the Vault. In this second scenario, I added the structure, but we see an ID because that structure is already registered in my Vault, but I can click register and register it as a new batch. And that's what I have in here. It's registered. Now we can see the molecule name and the batch number as well. Okay, so also very interesting is this insert link option. And you see here that I have different links, link into a molecule from a different Vault, link into some results or assay data. And this is all possible using this insert link function. This insert link function, in essence, it's a search function which allows you to search for compounds registered in your Vault or entities. So in this case, I'm on a small molecules Vault. If I type the prefix of the registration system, I get a drop down with all the molecules registered in this Vault, and then I can just select which one I want. But also if I just type a prefix of another Vault that I'm a member of, for example, AB is a prefix of the registration system of my antibody Vault, I can also view all the antibodies registered on my antibody Vault, and then maybe I can just select which one I want, and then I can actually link it with some other text, okay? So you can personalize this or specify what you want in that link. So this is possible because the link across Vault is enabled, and therefore you can just search for all the entities or molecules registered, not only in the Vault where you are working on, but also in other Vaults that you are a part of. Similarly, all the assay data can also be searchable. And here, if I just type for CRC, which is a protocol that I have in my Vault and stands for concentration response curve, I can see the protocol and also all the runs of that protocol. So it searches, you can search by keyword. Similarly, if you may be searching just for data, that was registered on a particular year. So maybe I'm searching for data registered last year. And here you can see that in this particular vault, we do have 3 runs. So it's a way of linking all the data, the structure side of your data, you can link it in your CDD, in your electronic lab notebook. And of course that when we do that, a link to the ELN experiment is automatically created. Okay, yeah, so chemical reactions and stoichiometry table, that's also available as well. I'm not going to spend time in here on this today, unless then if you have any questions. Of course, that Microsoft is fully integrated in the ELN, which means that I can drag and drop any Microsoft file, and then we can view the content of the file. The same, so either Excel or PowerPoint files, or any Microsoft file, you can add it in here. You can view the content. You can also open in Office 365, and you can even edit the file on the fly. If there is already, so in this particular case, this is data from a screening assay. And if I already have a protocol with the containers to store this data, I can simply click import the file, and this takes us into the import data tab, where I can register this assay data in the structure side of the vault. So this is very useful because you can be working in your electronic lab notebook following the ELN template, and then you can go ahead and register the data in the structure side of the vault directly from the ELN. Of course, that when we do that, we get the link to the particular experiment. Okay, also, as mentioned, sorry about the scrolling up and down. Let's just go here all the way down again. We talked about, or I mentioned assay annotation before. Just refresh here the page. The assay annotation, basically, let's just go all the way here. Sorry about that. Okay, there we go. So the assay annotation is this pencil shaped icon in here, and it's a useful tool to keep a summary of all the details of your assay. And this is basically, it is based on publicly available assays where all the terms were curated, and now all the terms are based on ontologies. So when we go here into edit mode, you see here that I chose all these terms, and depending on the term that I chose in the previous field, then we can see proposals which are related with the previous fields. And this is because it's based on ontologies. This is a useful tool, especially for traceability, because in 2 years down the line, you may want to check what instrument you used on a particular experiment or what assay kit. And you can simply come into the experiment and take a look. But also all these terms are fully searchable from the main ELN index page, as I will show you in a moment. Right. Also, many users use the ELN just for unstructured data. We've seen that the ELN, it's a flexible place where you can add and write in a flexible way. So if you have, PowerPoint slides from a research project or from weekly meetings, or if you have draft reports or scientific papers, or even if you have important emails about the research project, that can all be captured in the electronic lab notebook. If the relevant members belong into that project, they will all view or see the same thing. So it's one single source of truth. The advantage of having all this captured in the ELN, it's because when we go into the main ELN index page, so here I'm in my main ELN index page, I can search by keywords, right? So if I just, I can either type a keyword, I can also search by substructure, if I have any chemical reactions or structures in here, but I can also filter the experiments maybe by a specific author or creation date as well. So here, maybe I just want to view all the experiments created on a given period. So this allows to kind of filter your ELN experiments, searching by keyword and by substructure if you happen to have small molecules in that vault. So this is kind of course, that the Vault administrator can export all the ELN experiments, but also individual users, provided that they have export permissions, they will be able to also export one ELN experiment at a time in this button in here. Okay, so just a summary, we've been through the Explore Data tab, where we can register molecules, antibodies, ADCs, or any other types of entities. We've seen that we can capture the assay data in a protocol or assay. We took a look at electronic lab notebook. Finally, I would like to show you our visualization tool, which is the place where you can plot, filter, and mine the data. So in here, basically, I performed the query, and I have a search results table with similar to the ones that you saw earlier. But in this case, it has many columns of numerical data, some properties, and also assay data. When we have at least 2 columns of numerical data, we will see this option to launch visualization. So clicking the blue button, we get a new tab where we get a 2D scatter plot with 5 parameters. And here I can customize my x-axis, maybe to some other property. Maybe I want my % of mutant 1 inhibition. The y-axis, I can also assign it into any variable that I want. And then we have this settings wheel on the top left, where I can assign 3 more parameters. The color, maybe I want to assign it into the molecular weight. I can customize the range of colors that I want to use, and I can bin the colors based on the molecular weight. So maybe I can just customize this in here, and I can even add more bins and even change the colors for those bins. Oh, not that one, maybe this one. I can assign the shape into a different parameter, and also the size of the dots can be assigned into a different property. So maybe my log S. The opacity can be changed in here as well. And of course, because this vault is for small molecules, I have the structure registered. You can see that we get all the structures as I hover over the dots. Of course, that this is, we can zoom in into a particular area of interest by just dragging the mouse. But we also get all these filters on the right-hand side, where we can filter the data or simply add more filters and then just use these really nice sliders to just choose and filter the data that we want. Okay, also, so now you can see that I have 35 molecules selected out of the original 166. What I also really like in this visualization is the ability to color code the table. Clicking in this small arrow, it just maximizes the table that was transferred from the search results table. And then here I can color code the table based on some of the properties. So maybe I want to color code it based on the molecular weight, and I can just change these. These are the settings that I edited when I binned the molecular weight, but I can just override this if I want to. And then we can just go and see all the color codes table. Also, if you're working with small molecules, we have this integration here with SAR, with SuperSAR, which basically will show you all the fragments that are similar. So for this particular compound, we have all these number of molecules with that same R group. So that's quite useful as well. Okay, let's just minimize this table. From this actions menu, we can add another scatter plot. We can add an histogram, and we can add a bar chart. So we can have up to 4 plots all in the same page. They are all interconnected, and you can even see that as I slide through these bar charts, we can see the position of that particular compound in all the other fields. And of course, they can all be customizable. From the settings menu, there's one thing that I haven't mentioned yet, which is the tooltip. And basically the tooltip allows to decide what we want to, which properties I want to view in the card view. So basically, because this vault is for small molecules, I can pin a molecule by doing shift click, and I can just pin that particular molecule in that chart. And then the properties that appear in here can be customized in the tooltip. We also have a statistics tab, which allows to add a linear regression on the scatter plot. As we can see it in there, we can see the R-square and the slope. All these images can be saved as vector graphic publication images. So you can choose one of these options, or you can save as Excel. We can also add these 35 molecules into a collection. So this is another way of creating a collection of compounds which can be shared with the members of your team. Also, from this actions menu, you can use launch the session, or you can simply come into this URL. So you don't need to be a CDD Vault user. You can simply come into this URL, and then you can upload either an SD or an Excel SX file or a CSV file, and then you can plot the data points in the visualization tool and filter and mine your data. So, and with this is the visualization. I think I need to allow some time for questions, but just before that, I just would like to mention that CDD Vault also does have an API. So I'm just coming here into the help, into our knowledge base, which is quite a comprehensive knowledge base where you can see where you can find different sections. And then, of course, our API documentation is here. It's a RESTful API. There are some integrations already with CDD Vault, which you can use, and you can also view all the function calls. So if you want to register your entities or your assay data in an automated way, you can use the API for that. And with this, I think that is the end of this workshop. If there are any questions, let me just find the Zoom.
Thank you, Susana. Thank you very much indeed for that extremely comprehensive overview there. Really, really interesting. We do have some time for questions still. So for those online, please feel free to drop them into the chat, or if you'd prefer to use the raise hand function, as we found yesterday, some delegates were doing that, please feel free to do that as well. Just a question from me, Susana. Typically, how long, what would you say is the setup time for this software when you're working with a new client? How long does it take to kind of get it up and running?
Yeah, well, we would say that it is quite fast. So creating a vault basically by CD support takes usually 1-2 hours. Well, it's very easy to create a vault. But usually what we will do is we provide a demo for the user, and then we create a vault or a sandbox for the user, which is, it really is, it's very fast. So I think I would say that the setup time would be 1 to 2 days, and that's including the demo and the kickoff session. So CDD support will do a demonstration like we just had today, and also will help you, you know, kind of creating the own business rules and help you to register the entities, we basically train you on how to use the Vault. So I would say maximum 1-2 days, depending on the availability, of course.
Excellent. I assume then also there'd be sort of ongoing support as well, should any sort of queries and questions arise.
Yes, absolutely. Yes. So CDD support is available. Any questions can be sent into support@collaborativedirect.com, and then either myself or another member of the CDD support team will get back to the user. And usually, yeah, we are usually very fast, but that's the feedback that we get from the customers.